Gene therapy is rapidly emerging as a bold and potentially transformative avenue in the quest to slow or even reverse human aging. Among the most promising targets for intervention are telomeres, the protective caps at the ends of chromosomes that shorten each time a cell divides, and the intricate systems responsible for repairing DNA damage. In aging cells, both of these mechanisms degrade. Telomeres become critically short, triggering cellular senescence or apoptosis, while accumulated DNA damage compromises genomic integrity and drives dysfunction across tissues. Restoring these systems at the genetic level could address aging at its root, and early research in animal models strongly supports this possibility. In mouse studies, activating the enzyme telomerase, which rebuilds telomeres, has led to remarkable outcomes. In one experiment, researchers turned on telomerase in adult or aging mice and observed rejuvenation of multiple organs, improved metabolic health, and significant extensions in lifespan, without increasing cancer risk, which had long been a major concern. This was a turning point, demonstrating that telomere lengthening could be not only safe but powerfully beneficial when applied with precision. Since then, additional studies have reinforced these findings, showing that telomere maintenance can revitalize stem cells, improve tissue regeneration, and delay age-related disease. Building on this foundation, gene therapy platforms using viral vectors, synthetic mRNA, or CRISPR-Cas9 systems are now being developed to deliver telomerase directly to human cells. These therapies may be designed to activate telomerase only in specific tissues or under controlled conditions, minimizing the risk of uncontrolled cell growth. The real breakthrough will be combining therapeutic potency with safety, making sure that reactivated telomerase supports cellular health without tipping the balance toward malignancy. In the near future, early human trials may begin by targeting specific age-related diseases with known links to telomere attrition, such as idiopathic pulmonary fibrosis, aplastic anemia, or certain neurodegenerative disorders. If successful, broader applications aimed at healthy aging could follow. Alongside telomere therapies, there is growing interest in repairing DNA damage directly. CRISPR and related tools offer the tantalizing ability to fix mutations or reverse age-related epigenetic drift at the genomic level. One line of research focuses on enhancing the expression of key DNA repair enzymes, potentially making cells more resilient to oxidative stress and environmental insults that accelerate aging. Another strategy involves upregulating genes like SIRT6, known to play a critical role in maintaining genome stability and regulating inflammation. In animal studies, mice engineered to overexpress SIRT6 not only live longer but exhibit improved glucose metabolism and neurological function. These findings point to a future where a handful of gene switches might significantly alter the trajectory of biological aging. The key challenge in all of these approaches is precision. Aging is not a disease caused by a single mutation, but a complex, multifactorial process. Gene therapy must therefore strike a careful balance, enhancing protective mechanisms like telomerase and DNA repair without disturbing the finely tuned systems that prevent cancer and maintain immune surveillance. Yet advances in delivery technology, such as tissue-specific viral vectors and inducible gene expression systems, are making this level of control increasingly feasible. Tools like base editing and prime editing may allow for even more refined corrections, avoiding double-strand breaks and off-target effects. In the coming decade, it's likely that telomere lengthening therapies and DNA repair gene edits will be tested in humans, not as science fiction, but as cautious, well-regulated clinical experiments. The first generation of patients may be those suffering from accelerated aging syndromes or severe degenerative conditions where the benefits outweigh the risks. But as safety is established and techniques mature, the door could open to preventative applications, treatments designed not to fix disease, but to delay its onset by decades. Eventually, gene therapy for aging could become personalized. By sequencing an individual's genome, assessing telomere length, and analyzing DNA repair capacity, physicians may develop bespoke interventions tailored to a person's specific aging profile. These might include gene editing to repair accumulated somatic mutations, epigenetic rewiring to restore youthful gene expression, or telomerase activation cycles delivered every few years to maintain regenerative capacity. Combined with other longevity interventions like senolytics, reprogramming, or NAD+ restoration, gene therapy may one day form part of a comprehensive anti-aging protocol that operates at the deepest level of biology. While many hurdles remain, ethical, Technical, regulatory, and social, the trajectory is unmistakable. What was once considered too risky or speculative is now grounded in data, supported by animal models, and inching toward human application. If carefully developed and ethically deployed, gene therapies targeting telomeres and DNA repair could usher in a new era where aging is not simply accepted, but managed, delayed, and perhaps eventually overcome. The future of longevity is no longer limited to external interventions. It is moving inward, to the very code that governs how we age.